Next we have from Portugal, Zuzia Tomkielska. All right. And she is going to be speaking about their recent research that they did at the Azores University.
So, hello everyone. My name is Zuzana. I come from Poland, but currently I'm working at the University of the Azores, and I'm very happy to be here today with you and to share the research that we've done with our research group about the donkey milk. So, as we know, donkey milk has been traditionally used because of its nutritional and also therapeutical properties. The ancient Greeks and Romans, they used donkey milk also for medical purposes. And for example, Hippocrates, who is the father of modern medicine, he recommended donkey milk for a fever and also for digestive problems. So back then, people already knew that this milk has a positive effect in our health, but now we know what stands behind it. And it is already well known and described that this milk is a rich source of various vitamins, minerals, also bioactive compounds like, for example, immunoglobulins and lysozyme. And here on this graph, I mentioned some main biofunctional properties of this milk that I would like to focus on, which is antioxidant, anti-inflammatory, and antimicrobial activity. Donkey milk can also participate in immunomodulation and anti-aging. So, what exactly makes donkey milk so special? First fact is that donkey milk is very similar to human milk, especially in terms of proteins. Also, casein to whey protein ratio is very, very similar to human breast milk, and this ensures about two things. First is high digestibility of this milk, and second one is low allergic potential, because patients that have intolerance for cow milk, they are still able to tolerate donkey milk. So we did some research and we compared some components of this milk, of Asinus donkey milk with the human breast milk, and we noticed some very interesting facts, like, for example, donkey milk is a rich source of amino acids that have antioxidant activity, for example, like glutamic acid that can stimulate our immune system. Also, 2 times higher level of phenylalanine in Asinus donkey milk than in human breast milk. And this phenylalanine has antihypertensive properties. It means that it can treat high blood pressure and also can modulate the level of serotonin. We also noticed a high level of minerals, like, for example, calcium and phosphate. And we noticed one more very interesting thing, that is high content of iodine. And we can hypothesize that it might be due to 2 factors, where first one is the location, because I don't know if you know where the Azores is placed. This is an archipelago of 9 islands that are volcanic islands that are placed actually in the middle of the Atlantic Ocean. So this might be the first reason of this high iodine intake. And the second thing is the type of farming of donkeys, because diet of these donkeys is actually based on green and pastures of the grass. And now I would like to focus on these biofunctional properties that allow us to classify donkey milk as a functional food. And first thing is the presence of lysozyme. Here, what is interesting is this level of lysozyme is 6,000 times higher in donkey milk than in cow milk. Lysozyme is an enzyme that has antimicrobial properties and is present, for example, in our tears. And what is important for us is that lysozyme can modulate microbiota that are present in our guts. By saying modulate, I mean it can control the growth of harmful bacteria, while in the same time it can promote the growth of beneficial bacteria for us and for our health. And as we know, healthy guts means improved digestion, improved immune system, and also what is interesting, it's connected with our mental health. And when the barrier in our guts is compromised, it can lead to various diseases and disorders, like, for example, autoimmune diseases. So here what we did is we compared samples of milk collected from donkeys 2 months, 4 months, and 6 months after postpartum. And the last one, I'm not sure if you can see on this slide, is a commercial blend of Asinus. And as you can see here, the commercial blend, this milk has very, very strong antimicrobial activity. And this is the first thing. The other thing is that pasteurization and freeze-drying method that Asinus company developed had no significant impact in the presence of lysozyme. Also, the other biofunctional property of this milk is the antioxidant activity. So the antioxidant capacity of this milk is because of the presence of amino acids, vitamins, also enzymes like catalase, peroxidase. And these antioxidants are also very important for our health because they are able to fight against harmful molecules that are called free radicals. These free radicals, they can be produced in our bodies in natural way, for example, during metabolism, and also by some external factors like pollution, smoking, and radiation. And we again compared samples of milk that was collected from donkeys 2 months, 4 months, 6 months after postpartum. Then on the graph we have commercial blends of Asinus, and the last one is Trolox, which is analog of vitamin E that has very, very strong antioxidant activity, as you can see. And so the outcome is that this commercial blend has very, very strong antioxidant activity, as you can see, around 75%. And we calculated that 100 milliliters of this donkey milk is equal to more than 1,000 milligrams of Trolox and more than 700 milligrams of ascorbic acid, which makes a cup of donkey milk a way, way more stronger antioxidant than, for example, a cup of orange juice. And now I would like to focus for a moment in immunomodulation. And in particular, I would like to focus on immunoglobulins G that are present in this milk. So IgGs are being produced in our bodies as a response to presence of harmful pathogens such as bacteria or viruses. And they are, as you can see here on this graph, these antibodies, they have a site that is called antigen binding site. And by this specific region, they are able to recognize, bind, and neutralize these pathogens. And they are very important because people with IgG deficiency, they are more likely to get infections. So what we did is we compared the samples of raw milk, milk after pasteurization, and milk after freeze-drying process, and we noticed very high concentration of immunoglobulins, that is around 4 milligrams per milliliter. So even after very demanding processes like pasteurization and freeze-drying, this milk has still high concentration of these immunoglobulins. But one thing is the presence of these immunoglobulins, and another thing is that if they are actually active. So we decided to go a bit further with this assay, and we used 11 bacterial isolates, gram-positive and gram-negative, that you can see here in this table. And what we noticed is that all of the 11 bacterial isolates that we used were recognized and were bounded by these immunoglobulins found in donkey milk, which means that besides the fact that they are present in this milk, they are still active to perform their functions. And to understand better the impact of donkey milk in humans' health, we are using simple model such as Drosophila melanogaster, and it's commonly known as a fruit fly. So Drosophila share many genes and molecular pathways with humans, which makes it very useful to study. And moreover, Drosophila has also relatively simple genome, which makes it even easier to study this subject. So in this essay with Drosophila, in the first stage, we fed flies during the 5 following days with the donkey milk whey protein. Why whey protein? Is because Drosophila is not able to digest casein. After these 5 days, we started an immune challenge by doing injection of Escherichia coli, which is gram-negative bacteria. And we noticed some interesting results. That two results. That first one is the whey component, whey proteins of donkey milk increase response of these flies to this bacterial infection, and also that these flies' response were dose dependent. And we also used another molecular tool that is called GFP reporter gene, which is green fluorescence protein. And this is a very useful tool to visualize the genes or proteins of our interest. And here, what you can see in this slide, these are hemocytes that are immune factor cells. They are participating, for example, in phagocytosis, but they are also participating in inflammatory pathway that is called JAK-STAT pathway. So, in brief, when we have infection in our body, the cells of our body in this place of infection, they are starting to produce, in particular, hemocytes. They are starting to produce UPD3, which is pro-inflammatory interleukin. And this production of this UPD3 it's activating the transcription of genes that are involved in immune response. So we noticed that the flies that were fed with the whey protein, that they were able to produce a higher amount of this pro-inflammatory interleukin. And we also noticed that the flies that were fed with this whey component, they were able to faster recovery after this inflammation. So to sum up, we already discussed that donkey milk has very strong antimicrobial activity. Also, it's a very good antioxidant and can boost our immune system. But it has also big potential to be used in the cosmetic industry. So the ancient records says that Cleopatra, who was the last pharaoh of Egypt, that she used to take baths in the donkey milk to keep her skin young and soft. And we evaluated donkey milk by testing the inhibitory effect in collagenase, elastase, tyrosinase, and hyaluronidase. And we noticed significant inhibition in elastase, that is responsible for wrinkle formation, and also tyrosinase, that is responsible for hyperpigmentation. Hyperpigmentation, as you can see in the slide, this is a disorder that is characterized by black spots on the skin. And responsible for this is tyrosinase. And it occurred that whey proteins of donkey milk are able to inhibit both steps of both steps of tyrosinase activity, which is conversion of L-tyrosine and also conversion of L-DOPA. And we calculated that 1 milligram of donkey milk whey protein is enough to inhibit both of these conversions, and both of these conversions that are responsible for this hyperpigmentation. So thank you very much.